Hello, everybody, and welcome to today's webinar. My name's Melissa Burke, and I'm a Scientific Training Officer in the training team here at the EBI, and I'll be your host for today. So this is the third webinar in our series, Bringing Data to Life, a data management for the biomolecular sciences. So it's my pleasure to introduce our speakers for today, Kiva Cochrane, Nancy George, and Maria Levchenko. So Kiva is a technical officer within the metabolomics team at the EBI, where she manages curation and outreach. She has a degree in biochemistry and a PhD in molecular therapeutics, and has significant experience of working with small and large biological chemical data sets, having worked in drug development, R&D in academia and industry for many years. As part of her role in the metabolomics team, she supports submission of metabolomics data to the Metabolites database. Nancy is a scientific curator in the Gene Expression team, where she works on ArrayExpress and Expression Atlas. In her role as a curator, she works with scientists supporting the submission of gene expression data to ArrayExpress. Maria is the Europe PMC Community Manager, where Europe PMC is a literature repository and partner in PubMed Central International. She has a PhD in biochemistry from the University of Göttingen, and in her role as a community manager, she engages with all stakeholders of Europe PMC to communicate their feedback to the team of developers so that they can deliver the best tools for scholarly publishing and communication. So now I'm going to hand over to Nancy, Kiva, and Maria, and they're going to talk to you today about what happens to your data.
Hi, good afternoon. My name is Kiva Cochrane and I work for the Metabolites database at EBI. So in the previous webinar in this series, Ewan Birney and Elizabeth Bush addressed the ideas of altruistic and selfish reasons for storing data. Data storage is also becoming a prerequisite for publication in many journals, and data management plans are often required for funding applications. So great, now you have your data, what do you do with it? In this webinar, we will address how to store data and what to store. We will cover what we mean by data, principles behind storing data, how to store, find and reuse, data repositories available at the EBI, basic requirements in setting up a database, including standards for data repositories, repository examples, and a few frequently asked questions at the EBI. To start, let's look at what we understand by data and what it means in a biological context. Generally, we identify three forms of relevant scientific data: raw, processed, and metadata. But what do we mean by each of these terms? Raw data is the very basic information as it is captured, whether this is a photograph or an instrument file. Nothing has been done to alter this data. Processed data is raw data that has been altered. For example, the brightness of a photograph may be adjusted or the color intensity of part of an image increased to make it stand out. In biological data, processed data can include manipulation of the raw data, for example, normalization relative to blank controls or removal of outliers. It can also refer to post-analysis identification, for example, confirmation of a protein or chemical identity, comparison of expression levels, or statistical outputs. Metadata is the information relating to how the data was produced. For a photograph, this would include the make and model of the camera, the lens used to take the picture, post-processing software, and of course, who or where it is from. For biological data, we aim to capture as much information as possible. This will be everything from where the samples came from and how they are processed, which assays were used, potentially in as much detail as batch numbers for kits or plasticware if it's available, instrumentation specifications, through to which lab conducted the work and who the contributing authors were. Now let's look at where to store this data. There are two main types of databases: data repositories and knowledge bases. Data repositories aim to store, organize, validate, and make accessible core data. Each repository will have its own requirements to recreate a study. A comprehensive repository will store the raw, processed and metadata and all information essential to perform the experiment and analyse the data. This can be likened to an electronic lab notebook. Knowledge bases are generally resources which provide curated collections of subject-specific information. For example, a collection of cross-omic information, which might link a gene transcript variant with its corresponding protein and reactions in which it's involved in within a species or group of species. These can be likened to encyclopedias for the biological sciences. Data repositories can provide the evidence base for knowledge bases. When contemplating which data repositories to use, it is important to think about what you're storing and what the purpose of this is. If data is to be open source and easily accessible by your community, it may be advisable to use a subject-specific database. These often benefit from also being structured to prompt for specific or essential information related to the field. Journals which require data deposition often provide lists of recommended repositories. Consideration should also be given to storage capacity. Do you have the ability to store in-house, or would a free service be a better option? Manually curated repositories also come with the added benefit of a direct line of support for submissions and a knowledge that all submissions meet the minimum acceptable requirements. At EMBL EBI, we offer various resources to support the multiple different data types, all under one roof. An overview of these can be found via the URL on the screen. If you're unsure which database is suitable for your data, please take a look at the EBI Data Submission Wizard. This provides a step-by-step guide to help you identify the type of data and which of our resources matches your needs. Data storage has to have a purpose. Generally, that is to be able to find the information again, whether that's internally within a company or university or publicly within open source resources such as those at EBI. With the ideas of data sharing has emerged a need to improve the infrastructure supporting the sharing and reuse of scholarly data. Some of you may be aware of the FAIR data principles, which were developed by a diverse set of stakeholders representing academia, industry, funding agencies, and scholarly publishers. The main outcome being the data should be fair, that is, findable. Data should be associated with its unique identifier. Accessible, the information should be searchable by man or machine. Interoperable, it should be controlled vocabularies, that is, a formal, accessible, shared, and broadly applicable language. And it should be reusable. Data should be associated with its provenance. FAIR has formalized many of the processes which are the foundations for EBI databases, which we look at in this and following webinars. The FAIR principles are becoming a more significant feature in both funding applications and journal submissions. Submitting to data repositories such as those at the EBI can help to meet these requirements.
So this is where I come in. So I'm Nancy. Nice to meet you all. So going back to setting up a database based on FAIR principles, once a community has decided it needs to track and store the data, it comes the issue of what's the best way to go about this whilst taking into account community standards. So these are two examples of specialized repositories available at the EBI. The first being ArrayExpress, which would be the database for microarray and RNA sequencing data, and the second one would be Metabolites. So the requirements were for open access, subject-specific storage for raw data, and the data also needs to be easily accessible, readily searchable, and these can be done using structured formats, ontologies, control vocabularies, and this also allows entities to be searchable by both man and machine. And what formats would be most effective for this, and how will this be used? So, setting up standards are usually done by shareholders in the community. So for sequencing and functional genomics data, this would be the standards we use here from the Functional Genomics Data Society, which was established in 1999. And these are advocates for open access to genomic data sets, and they provide standards on how to do this. For example, this is where the Miami and MNCICI minimum information about a microarray or experiment or minimum information about a sequencing experiment come in. And on the metabolite side, this would be the Metabolites Standards Initiative, which was set up in 2007. And these provide guidelines on by the Metabolite Society. And early efforts were focused on community agreed reporting standards, the so-called minimum information checklists and data exchange formats. And this was their aim was to provide a clear description of the biological system studied and all the components of the metabolomics study, as well as how to allow data to be efficiently applied, shared, and reused. So my role here at the EBI is with both Annotaire and ArrayExpress. So functional genomics data at the EBI is submitted through the web tool Annotaire and displayed at the database ArrayExpress. So Annotaire is the web submission tool. This is essentially a fill in the boxes step-by-step guide to submitting functional genomics data. And the outcome is 2 files. So the first file we call the investigation description format. And this basically captures the experiment information. For example, who is the submitter? Where are you coming from? Basically, what's your institution? Which submitter did what? So if you have multiple people associated with the data set, then you can capture that sort of information. Are there any publications pre or post accepted for the data set? The second data file we produce would be the SDRF, and this we call the sample and data relationship format. And this captures the progression of the sample from the biological material all the way through to what protocols were used, which protocols apply to which samples, what were the protocols used, a short description of each protocol. And then sample annotation is linked to control vocabularies, what we call ontologies, and these will be covered in more detail in the next webinar. And this allows us to help submitters annotate the metadata to clearly describe the metadata and to allow cross-experiment comparisons of the data. So basically, Annotaire also allows you to capture the raw data as well. So we allow users to upload the raw and processed data and assign which files go to which samples. And here we also capture things such as technical information about the files. For example, for sequencing experiments, this includes library strand information, whether the library is paired or single end, and the file to sample relationship. For example, if there are multiple files for each submission, whether you've got more than one file, for example, for a paired library, there would be 2 files per sample. Or if you have technical replicates, there would be many more files per technical replicate. And if you have processed data, you can assign those to the samples too. So once a submission is submitted by the user, we run checks. The first is to make sure, a metadata check, to make sure that all the information about the samples has been provided. And the second check would be a file integrity, file validation check. So if there were any issues uploading the file, to make sure the file isn't broken, and to make sure that all the files that are required for each sample are present. And once those checks are done, the submission is complete and they get given a stable accession number, which can be used to find and cite the dataset. So once the submission has been provided by the user, that's where the curators come in. And we also take a look at the submission to make sure all the information makes sense and to make sure that there isn't any missing information. For example, if you have a submission where you're missing one of the protocols or the protocols aren't clear, we come back and we make sure that, you know, this gets sorted. Or if there has been a mistake in the sample annotation, for example, if your sample is labeled male, but the organism part is something like endometrium, then that generally raises a flag. And once we've ensured that all the data and files are correct, this then gets processed and uploaded to ArrayExpress, which is the database here at EBI. We also, so here, this is an example of an uploaded dataset. So you can see it gets an accession. Here is emtab. And then from this page, you can look at the sample to data relationship format. You can look at the protocol information, who submitted the data, and you can also find things like how to contact the submitter and also download the data that's highlighted in the green box. So the functional genomics raw data is stored at the European Nucleotide Archive, the ENA, which is part of the Sequence Read Archive, a collaboration for functional genomics data between the ENA here at the EBI, GenBank at the NCBI, and DDBJ in Japan. And microarray raw data is stored here at the EBI as well.
So a second example of a database is the metabolites database, which is a repository specifically for metabolomics data. The requirements for data submission have been guided by the Metabolomics Standards Initiative, the community standards mentioned previously. Metabolites is a fully curated repository which requires a minimum submission of raw and metadata. So what does a metabolite study look like then? In keeping with the FAIR principles, metabolites provides each study with a unique and persistent accession number to ensure it is findable. We also link studies to any associated publications. Public studies are fully accessible with information stored in a searchable manner. For example, the protocol information, where not only are the methodologies provided, but the specific information, instrument information is highlighted in a manner readily searchable by man or machine. We collate as much information as possible surrounding the provenance of samples and experimental procedures. So sample names, treatment conditions, analysis run orders, all of which helps with the reuse of the data. Controlled vocabularies, as can be seen here in the example with organism, mean that metabolite studies are also interoperable. Metabolites collects the raw data files and where available processed files as well. This includes the all-important metabolites identified in the study, which are then linked back to CHEBI, the chemistry ontology, to help to standardize the nomenclature for metabolites and improve interoperability. And to ensure metabolite studies meet these criteria, there are both automated and manual validation processes in place. And at the end of the process, studies are open access and can be easily downloaded for reanalysis. So just to finish off this section, we wanted to talk about a few frequently asked questions that we get here at the EBI. So to start off, just a little bit about file storage, the where and how. At EBI, whether you have a small or a large amount of data, we have the storage capacity for your data. And if your data is too large for normal upload and download methods, there is also alternative options available through, for example, FTP folders and Aspera, which will be available through each of the different databases. What about identifiable data? Any data that is protected can be submitted via the European Genome Phenome Archive, provided the correct permissions are in place in terms of ethics. EGA provides a controlled access service, meaning data will only be released following a strict permission protocol. Often submitters don't come to databases until they are just about to publish. And often this is at a panic stage, as the publication cannot go through unless the data is submitted. The reason for leaving submissions so late is regularly contributed to concerns of data being made public before the publication is actually released. We want to assure submitters that we have controls in place at EBI that prevent your data becoming public until a date that is agreed upon by the submitter and the repository. For example, at Metabolites, your study will be private until the end of the process. Prior to this, once your study has passed the curation stage, if you wanted to share it with a collaborator, or if a journal reviewer requests evidence of your raw data, we can supply you with a link which allows you to share this with the parties concerned, so that they can view your study, although they will not have editing rights. And finally, do not fear, we do have user guides and we have contact support available for the repositories.
And with this, I take over. Hello, everyone. This is Maria. And today we have talked a bit about how to submit your data and where. And we've also explored the question of why you would make your data open in the previous webinar. But what is it that happens after you submit your data? And this is the part where I'm going to talk about credit. As a researcher submitting the data that I have generated, I would like to know, once my data is in a public repository, who owns it? What are conditions for others to reuse it? How can I cite it in my own research? And will others give me due credit? So what's in it for me? So starting with the first question, who actually owns the data before and after its submission? And I've just put here the EBI terms of use for the online services and databases. And as you can see on the slide, for all the data that is generated and contributed by the community, it is the community that remains the data owner. It's also important to note that public databases at EBI place no additional restrictions on the use of sharing of such data other than those that are specified by the original data owners. So ownership is a slightly different concept from copyright though. At many public repositories, deposited data is placed in the public domain. What is public domain? Well, public domain consists of all creative works to which no exclusive intellectual property rights apply. And here I have an example from Protein Data Bank, and you can see that all primary and derived data are placed in the public domain once they appear in the repository. However, I just wanted to highlight that this doesn't mean that you lose control of your data once you deposit it. Coming back to the EBI's terms of use, you can see that the original data may have had some restrictions. As mentioned before, it might be sensitive patient data, or it even might be protected by a patent. And all these cases are taken into account upon submission, so you don't have to worry about data misuse. Some public repositories we've seen, as Zenodo and Figshare mentioned briefly, can also offer you to choose a license. On this slide, I've put several examples of different Creative Commons licenses, but there are many more different types. And each of the license types comes with its own terms. Some may request attribution, some may prevent commercial use, and some may prohibit derivative works. Now, it's up to you which license you would like to choose. But I'd like to share a quote from Henry Ford. He said, if I asked people what they wanted, they would have said faster horses. It's hard to predict innovation. Back then, cars were something inconceivable. And similarly, this happens with scientific works. I work for a literature repository, Europe PMC, and licensing information for research publications determines how we may or may not use and display them. For example, we and others text mine publications to identify useful facts. Here on the slide, I have an example where you can see that we found a protein-protein interaction mentioned in the text. Normally, you can highlight such evidence in the paper using a tool that we have developed. But if the license specifically prohibits derivative works, then highlighting is considered a derivative. So therefore, we can't do that. This is just something to keep in mind when you're selecting a license and just thinking that if you pick a more prohibitive one, it might actually prevent somebody from doing something useful with your data. Another important issue to address when talking about what happens to your data is, of course, a question of credit. How do you get credit for your work? In academic system, most often we give credit in the form of citations. Other researchers that build on your work will reference your input by citing your publications. So probably in a typical workflow, you generate data sets that are then described in publications. Others discover the data that you've published as articles, they read them, and then they might reuse or reanalyze the data, and that will again be described in another publication that will acknowledge your work in a form of citation. While this workflow is very common, it is by no means the only possible setup. And I have a couple of other examples that may happen. Some data never gets published, but others may nevertheless reuse and cite it. There are some large data sets that may be described in a number of articles. How do you reference a single data set? A single article may in turn contain a variety of data. In that case, how would you reference a specific data set? Or there may be different authors that have contributed their data to the paper. Is it possible to get individual credit for data in such a case? So all these use cases actually require that we sort of uncouple underlying data from the publication so that a data set becomes a unique, independent research object in its own right and can thus be reused and cited. Of course, if we separate the two, we also need a way to link them so that we know the full backstory for both the data and the publication. And for data to be used on its own outside of the publication context, it needs a unique identifier. You can think about it like a social security number, but for data. The unique ID is assigned or given to the data when you submit it to the repository. And different repositories may use different ID types. Think of it as barcodes versus QR codes. So the data identifiers have been mentioned previously in the context of FAIR data. And this is very important for us because it allows us to identify data specifically and it makes it findable. So sometimes data is given an accession number. For example, a protein structure of the Drosophila globin, which is a fly protein, is given an accession 2BK9 in the PDB database. If you search PDB for 2BK9, you will retrieve this particular data set. Some accession numbers may be designed in such a way that they make it easy to identify the data repository. So for example, proteomic data deposited in PRIDE database will always have accession numbers starting with PDX, like PDX and a string of numbers. But sometimes you only have an accession number at hand and it might be difficult to find out which database has issued it, like the 2BK9 example in the previous slide. So if you want to refer to a data set by its accession number, it's a good practice to cite the database along with it so that you mention which database this accession number belongs to. It's also good scientific practice to actually give credit to the database that stores the data. So you can see in this example that not only did the accession number and the database mentioned in the article, they have also cited the paper that actually describes the database itself in their references. Now, another type of unique ID for data is a DOI. It stands for Digital Object Identifier, and it works as a link. A DOI points or resolves to metadata describing the object in question. So in our case, most often, the DOI will be linked to the URL of the data record in a repository. The URL itself may change, but the DOI will always stay the same throughout the lifetime of the object. It is really stable. And a DOI looks like this. It will have a resolver service saying that it is a DOI. It will have a prefix that tells you who gave this DOI to the dataset, and a suffix which identifies what is it that you're giving the DOI for, what kind of data is this. So having these unique IDs for data allows us to refer to data on its own, meaning that you can cite the data, not only the article. And there may be different citation formats for data, but I've just put a minimal recommended citation format on this slide. It probably should include the following. We would really want to see authors, year, title of the data set, repository, potentially a version, and of course the identifier. Now, some databases will also give you guidelines on how to cite data. Here I have one example from ArrayExpress, and it actually describes what you should put in when citing a particular data set. Now, scientific journals where you submit publications to will also have often data policies. They will tell you where the data should be deposited and how it should be referred to in the publication. In this particular case, it's an example from Nature Journal. And as you can see on the slide, it is a requirement for publishing your research in a Nature Journal to make materials, data, code, and associated protocols available to readers without undue qualifications. And the journal might also specify what is their preferred way that you share data sets. And in this case, they suggest that you share it via public repositories, and they may even give you guidelines as to which repositories to use for which data. It also might tell you how you cite the data, so all the things that we went through in the previous slides. As you can see here, all manuscripts for Nature journals must include a data availability statement. This is the part that describes where can readers get the data that has been reported in this publication. And it is also important to note that supporting data must be made available to editors and reviewers at the time of submissions. So as Kiva mentioned, you don't have to wait all the way until the publication is released to put your data in a public repository. On the contrary, putting it in early ensures that you will have everything in place for a smooth submission at the journal as well. Now, perhaps you might be wondering whether all this actually is implemented in the scientific community. So do others actually cite data? And you will find that it is a very common practice. This is something that is a community norm by now. I've pulled up a couple of examples here. So in this case, apart from references section in the publication, you will also have a data citation section, which will list all links to the data. Or it might be part of the references. Here, if you look closely at the items number 19 and 20, this is a list of references, so it cites some papers, but some of them are actually scripts that have been cited just alongside the articles. Now, this, of course, is really great for credit, but it's also useful in a very different way. It allows you to search for specific data that has been cited in publications. And here I have an example for how data is displayed in Europe PMC. So you can see that in addition to the article abstract, you can go to the data tab that will describe all the data that has been cited in the article. So here you see there's a clinical trial and a proteomics data set that have been actually mentioned within the publication itself. So these things become visible and they're actually picked up by the community. And again, proper data citation actually enables better search tools that you may use. So again, if you would like to search for publications that, for example, reference some specific types of data, let's say you wanted to find all articles on a specific metabolic pathway that have a protein structure available, you could do that. This is a very specific requirement, of course, but it really allows us to find the types of data that you may be looking for. So this is one example. You can search for PDB accession numbers, and then in the publication, we can text mine them out when the text is available and highlight them for you. Again, on the subject of text mining, ontologies came up a couple of times, and they will indeed be described better in the next webinar. So I'd really recommend that you tune into that one as well. But it is really crucial that you properly cite all things, not just accession numbers, but sometimes it even comes down to the protein names. It is very difficult to identify, for a machine especially, to identify that this is indeed a protein, because we tend to refer them by common names. It's especially true for Drosophila science, where a lot of them might be very similar to commonly used words. Now that we've talked a bit about how to cite, let's briefly touch on the subject of credit. And here I would like to highlight ORCID IDs. Now, ORCID IDs are unique identifiers for researchers. They've been established initially to differentiate researchers with common names. If you're Jane Smith, it may be difficult when you're, as an author publishing articles, to know that these articles belong to you and not to Jane Smith that works in a different field. People also tend to change names, so all these kinds of things, in order to account for them, we needed a way to tie all your outputs to you as a researcher. And it is really easy to get an ORCID ID. You just need to register with a nonprofit organization, you add your information, and then you use your ORCID ID in grant reportings or when submitting publications, and so on. And this is how an ORCID profile looks like. This one is for me, so you can add your works. In this case, these are my publications, but also employment and education, or even grant information to it if you wish. And more importantly, now you can also add data. In fact, when you submit data to databases, and here is an example for metabolites, you can actually claim it to your ORCID. And so that becomes part of your own record, and you can cite it, you can show it, it will sort of tie the data to you as an author. Now, we've had some frequently asked questions that I've just bundled on this slide, and we've actually covered many of them. So we talked a bit about data ownership, and I've shown you how you can link data sets to your ORCID ID. But then it's also often asked whether you can link to your data set to a publication or perhaps even a preprint that appears in a repository like bioRxiv. And actually, in fact, it is also possible. So it doesn't have to be a journal publication. You could, when submitting your data, you could also tie it to a preprint. You can also link different data sets across resources, for example, related experiment links for other databases at EBI. And so it's really possible to create a whole landscape of connected data, publications, and authors that create these things. And we think that by bringing all these together, we can actually do science more efficiently. Now, with this, we're coming to the end of the webinar. I will just highlight that there is a next webinar coming up, and you can see and register for it via the link on this slide. And with that, we leave time for questions. So if you have any questions, you can submit them in the chat now. Otherwise, it seems like we've done a great job of making everything clear, or we've completely confused you and you don't really know what to think. In either case, we will be happy to answer anything that you may have, and you can also contact us later. We've put our contact details on the slides as well. And we would really appreciate your feedback. So when the webinar finishes, you'll get a feedback link. So please tell us what you think so that we can add this to the next webinars. Well, thank you very much. Well, it was absolutely great having you here. All the best and see you at the next webinar.